I'd like to thank organizers for giving me this opportunity to introduce our study of emergence and evolution of antimicrobial resistance genes in Neisseria gonorrhea, which was recently published in Genome Medicine. The resistance of Neisseria gonorrhea to third-generation cephalosporins or 3GCs has been defined as high priority by WHO. Strains from two internationally circulating STs are of great interest because they have acquired the mosaic PENA alleles determining the resistance. The two STs, ST1901 and ST7363, and mosaic PENA alleles likely originated in Japan. Previously, I reviewed different evolutionary paths of the two major STs, in particular in terms of distribution of two types of mosaic PENA alleles, PEN34 and PEN10, in which the second half of their DNA sequences appear to have been imported from other Neisseria species that are naturally resistant to 3GCs. However, the timeline, mechanism, and process of emergence and spread of the alleles to other countries remain unknown. Here, we obtained complete closed genome sequences of 3 Japanese ST1901 isolates harboring PEN34 from 2005 and reconstructed updated phylogeny of these genomes and 1,072 other genomes from isolates sampled from 35 countries. Regarding ST7363, we also sequenced the genomes of 103 Japanese isolates from 1996 and 2005 and reconstructed updated phylogeny of these genomes and 88 other Japanese genomes dating from 1996 to 2015. These large-scale dated phylogenies were for the first time inferred using backdating. Here you see a clonal dated phylogeny of ST1901 inferred for the approximately 1,000 isolates sampled from 35 countries across 5 continents. Most strains were isolated from North America, colored in light pink, and Europe, colored in pink, mainly through 2 large surveillance projects. Strains isolated from Asia are colored in blue. ST1901 and its single locus variants are colored in the ST color. The other minor S strains not colored in this ST column comprised 21 STs, but they were all classified as the same core genome group defined in multi-median lab based on core genome allele profiles. Next, we added information of resistant and susceptible categories to third-generation cephalosporins, cefixime and ceftriaxone, and presence of any mosaic PENA allele colored in cyan, and that of any non-mosaic PENA allele colored in green, light green. In general, isolates harboring the mosaic PENA alleles and others are separated into 2 clusters. Breakdown of the mosaic PENA alleles shows the majority is PEN34. The cluster at the top accounts for the majority of isolates harboring PEN34 and its variants, and hereafter it is named as sublineage 34. In this sublineage, about half of the strains were LNG must bound for 0 cell. Origin of this sublineage is indicated as the red circle and was estimated between May 1990 and July 1999. Meanwhile, breakdown of the non-mosaic alleles shows majority is PEN5. Most isolates harboring the non-mosaic PEN5 were located in sublineage at the bottom. Other minor isolates harboring PEN5 were present immediately outside the sublineage 34, suggesting PEN5 was ancestral and that PEN34 evolved from it. In sublineage 34, strains with the oldest isolation date were from 2005, among which 3 were from Japan and 2 from the USA. As I mentioned in introduction, we obtained complete genome sequences of the 3 Japanese strains isolated in 2005, and their phylogenetic location is indicated as the blue bar. They were phylogenetically located at the base of the sublineage 34 tree, and none of them were likely closely related to the ancestor of the strains that spread to other countries. To provide an outgroup for the sublineage study 4, we also obtained the complete genome sequence of a Japanese strain isolated in 2000, which is indicated as the green bar. This isolate harbors a non-mosaic pen allele and is phylogenetically close to the sublineage 34. For the newly sequenced complete genomes of the 4 isolates and a publicly available reference isolate indicated as the pink bar in sublineage 34, we conducted genome alignment, which showed conserved genomic regions among which the isolate harboring a pen non-mosaic pen allele and the reference isolate in the sublineage 34 and one of the isolates harboring pen 34 isolated in 2005. This indicated that the overall genomic structure was maintained during evolution from the ancestor of the isolates in the sublineage 34 that spread to other countries. When I again look at the entire phylogeny of ST1901 associated cogenome group in terms of mosaic pen 10 allele, you see 3 separate pen 10 allele acquisitions. One of them was phylogenetically close to sublineage 34 and included the oldest isolates harboring pen 10 from 2000 to 2001 in Japan. This result suggests that the pen 34 was generated via pen 10 from the ancestral pen 5 allele. Pen 34 and pen 10 are identical except for its 3' terminus. Next, I'd like to talk about the ST7363. The dated phylogeny showed that the isolates were broadly separated into 2 clusters corresponding to the absence or presence of non-mosaic pen allele. In contrast to the ST1901 associated cogenome group, we observed that the majority of the ancestral non-mosaic pen allele was pen 2, which is a pen 5 variant and has the same nucleotide sequence with pen 5 except for its 3' terminus. The majority of the mosaic pen A was pen 10, with the exception of a historical sporadic isolate resistant to self-reaction and one isolate harboring pen 34. The dated phylogeny showed that pen 10 in ST7363 was generated from the ancestral pen 2 between September 1991 and May 1995. To further analyze the origins of mosaic pen 34 and pen 10, we compared their nucleotide sequences to those of the susceptible alleles, including their downstream sequences, as shown in this schematic depiction. In ST7363, wild-type pen 2 and mosaic pen 10 are identical at the 5' region, followed by the recombined mosaic region, which is most parsimoniously explained by a recombination event, although the source of this DNF segment is unknown. In ST1901, wild-type pen 5 and mosaic pen 1034 are identical at the 5' region, followed by the recombined mosaic region. The coding sequence of pen 10 and pen 34 were identical, except for the 3' end. In the following region 3, the downstream sequences of pen 5 and pen 10 were identical, whereas those of pen 10 and pen 34 were not. In the following region 4, the nucleotide sequences of pen 5, 10, and 34 exhibited equal to or higher than 99.9 sequence identity. Similarly to ST7363, the origin of pen 10 was most parsimoniously explained by recombination event, although the source is unknown. Pen 34 and pen 10 differed in the 3' terminal region, and the difference continued in their downstream nucleotide sequences in the region 2 and region 3. To investigate the possibility that PAN34 emerged via recombination from PAN10, we aligned the nucleotide sequence corresponding to the orange region to genome sequences of these two sets of Japanese strains. An ML tree constructed from this alignment reveals that ST190 isolate encoding PAN34 and its variant were clustered with ST1594 strains encoding PAN1. It suggests a second recombination event from PAN1 into PAN10 background, resulting in PAN34. In summary, the origin of PAN34 in ST1901 can be explained by two recombination events. First, similar to ST7363, a recombination event from a commensal Neisseria into a wild-type PAN5 background, resulting in PAN10. Second, a recombination event from PAN1 into the PAN10 background, resulting in PAN34. In a published paper, I also analyzed susceptibility to fluoroquinolones and antibiotic use, but today I don't talk about them because of time limitation. I'd like to thank my co-authors, in particular, Jonathan Grad Lab and Marty Maiden Lab, for this great international collaboration. I also thank Xavier for instruction on backdating and supercomputers in National Institute of Genetics in Japan. Thank you very much for your attention.